Our recent discovery of a novel swine enteric alphacoronavirus called SeACOVICH in the southern China. This study was actually conducted by my lab at Zhejiang University and also collaborated with the hog production divisions at Guangdong Wang's Foodstuffs Group Cooperations. So the outline of my presentation will include 3 parts. First is the background introduction. The second will be the discovery of the SeACOVICH in southern China. The results of this part has been published recently in Veterinary Microbiology. And finally, I would like to share some unpublished update data on the SeACOVICH and give some summary. So as we all know, since December 2010, large-scale outbreaks of diarrhea with 80 to 100% morbidity and 50 to 90% mortality in suckling piglets were occurred in China and causing a very severe economic loss in China pig industry. And PEDV is the main pathogen responsible for these outbreaks. However, the other porcine coronavirus like TGV, PDCV, and the other unknown factors like torovirus, rotavirus may also involve in these outbreaks. So far, 4 porcine coronavirus has been discovered, TGV and PEDV. TGV and PEDV which belongs to alphacoronavirus, PHEV belonging to the beta coronavirus genus, and the PDCV belonging to the delta coronavirus genus. TGV, PEDV, and PDCOV are classified into the swine enteric coronavirus called SeACOVICH. Most recently, in Europe, TGV, PEDV chimeric SeACOVICH was also identified, probably due to the recombination between TGV and PEDV. So the alpha and the beta coronavirus genus was is believed to origin from the bat coronavirus, whereas the gamma and the delta coronavirus is believed to have derived from the bird coronavirus. And as a supporting evidence, previously we saw that the 5' UTR untranslated region of the emergent PEDV is more closely related to a bat coronavirus called BTCOV 512 identified in southern China. As you can see in this figure, position 1, 2, and 3. At these 3 positions, the emergent PEDV has the same deletion and insertion with the bat coronavirus 512. However, if the difference between the difference in this region is associated with porcine diarrhea, it remains unknown. So the PEDV-like bat coronavirus 512 was located in the southern China in 6 provinces, including Guangdong, Guangxi, Hainan, Jiangxi, Anhui, and Yunnan. At the same time, the research group in Hong Kong University also identified another alpha bat coronavirus called HKU2. That's why they give the name, because it's from the Hong Kong University. So this virus was identified in rhinoceros synacates, also called Chinese horseshoe bats. And this is the virus is not isolated yet. However, 4 full-length HKU2 sequence, 1 from Guangdong and 3 from Hong Kong, was reported. And the HKU2 RNA was detected in the feces of the healthy bats, indicating that it's enteric tropism. And the HKU2 S protein has a unique feature. It's the smallest S protein among all the coronavirus and phylogenetically related to the beta coronavirus. Also, co-infections of HKU2 alpha coronavirus and SARS-like beta coronavirus was detected in 1 animal. And most recently, the other newly identified rodent or house-flu coronavirus in China, similar to the bat coronavirus HKU2, also harbor the S genes divergent from the alpha coronavirus, as you can see in the red arrow. this group of the virus are classified into a separate clade, far from the alpha coronavirus, but close related to the beta coronavirus. So the HKU2-like bat coronavirus also can be identified in USA. This paper showed that the HKU-like beta bat coronavirus was identified in the big brown bat in Maryland. So this table shows the pool number 1 is used for the metagenomic sequencing analysis. So the results show that a total of 45 sequence reads derived from the 454 sequencing of the pool 1 samples was assembled to the bat coronavirus HKU2 genome, and then a PCR amplification of 2.5 kD fragment of OF1AB, the black fragment, which is called ARCOV, was used for the phylogenetic analysis. The results show that, as you can see here, the ARCOV OF1AB fragment is closely related to the HKU2 at the S gene nucleotide and the protein levels. So, if the virus is not jumped from the bat to pigs, we may not care about it. However, it did happen. So, beginning from the February 2007, in the Guangdong province, 4 nearby pig farms in the northern Guangdong province reported increased outbreaks of diarrhea in the newborn pigs, and the clinical symptom was indistinguished from PEDV infection. However, all the swine coronavirus RNA detection was negative, and also other known porcine pathogens detection was also negative. So subsequently, we used a pan-coronavirus RT-PCR assays targeting the RT-RP of all the coronavirus members we successfully detect, amplify 251 pp in the OF1B gene from the submitted samples. And the sequencing results show that all the samples were 100% identical to the nucleic positions, 14,000 and 24 to 14,274 of the bat coronavirus HKU2. These results implicated that the HKU2-like coronavirus was responsible for the outbreaks in the pig farms in Guangdong province. So the most important thing is to isolate the virus. We are very lucky just in the passage 2 in viral cells in the presence of trypsin. We can observe the cytopathic effects, CPE, characterized by the syncytin formation. So the virus has been, so far the virus has been passaged for 20 times, and by the RT-PCR detection, the virus is revealed to be the HKU2-like porcine coronavirus, which we call SeACOV. So we also have the serological evidence, as we saw that in these pictures. The positives and the negative cell serum can be screened in the virus-infected cells by the IFA. And we can also observe the virus particles by the electron microscopy, showing 100 and 120 nanometer in diameter, and with the surface projection typical of coronavirus. So we also saw that the SeACOV did not cross-react with the PEDV, TGV, or PDCV by using anti-nucleic capsid antibodies of each known coronavirus, as you can see here. And this is a one-way test. And recently, we also performed a two-way test. We generated SeACOV anti-nucleotide polyclonal antibodies in the rabbit, and we found that SeACOV antiserum nor the anti-nucleotide antibodies not cross-react with the PEDV, TGV, or PDCV infections in vitro. So this is a two-way test. They don't have any cross-reactions. So next, we genomic cloning of this screen by RT-PCR amplification of 15 fragments covering the full-length genome. Sequencing results show that the SeACOV genome has 6 nucleotides longer than the HKU2. This including 3 insertions and deletions at 3 positions, NXP3, S1, and M genes. So the SeACOV genomic sequence was about 95% nucleotide identity with HKU2. The most variable region was located at the N-terminal region, NTD. There are 75 amino acid substitution plus 2 amino acid insertion in the NTD compared to the HKU2. And in contrast, only 78 amino acid substitutions in the rest part of the S proteins. The phylogenetic analysis showed that the SeACOV together with the 4 HKU2 bad coronavirus HKU2 sequence were classified into alphacoronavirus at the full-length genome level. However, at the S gene levels, these 5 virus was clustered in separate clade, closely related to the beta coronavirus. So we also perform a structure-based S protein sequence alignment with the S proteins from the alpha coronavirus NL63 and the beta coronavirus MHV, because most recently the S proteins of this, the structure of the S protein of this virus has been resolved. So the results show that the NTD of SeACOV S protein is structurally resembled to the domain 0 of the alpha coronavirus, whereas the rest part of the S protein of SeACOV structure similar to the beta coronavirus. So the results reveal that the SeACOV S protein has a unique S protein structure, hybrid structure, comprising the alpha coronavirus and the beta coronavirus S. So during we preparing the manuscript, another group in China, in Guangdong province, also reported the study. They report describing the SeACOV sequence called GDS04 strings. And this paper published in the EID, and they have the similar phylogenetic analysis results. So actually, the source of this group was the same as us. So the company, after the outbreaks, the company distributed the same samples to different labs to help to identify the unknown pathogens. And the GDS04 was 99.8% nucleotide identity by the next generation sequencing with our string GD01. And these 2 viruses could have the same origin based on the sequence comparison. So to test whether the SeACOV is infectious in pigs, we conducted a pilot challenge study. 10 pigs, 10 3-day-old piglets with 5 pigs per group were assigned. And they were all infected with the SeACOV or the DMEM respectively. 2 pigs in each group were necropsied at 3 days post-infection, and the remaining 3 pigs were necropsied at 5 days post-infections. We performed fecal RNA detection per day and histological examination in the small intestine in the necropsied pigs because the SeACOV structural protein has not been available at that time. So the results show that we observed clinical signs characterized by the acute vomiting and watery diarrhea. However, the signs was not severe compared to TB challenge. And we also detected fecal virus shedding started at 27 to 40 hours post-infection and lasted thereafter. And gradual atrophy with a significantly reduced VH-CD virus can be observed in the jejunum and the ileum samples. However, it's not detected in the duodenum samples. So the final part. So recently we generated anti-SeACov nucleic capsid protein polyclonal antibody in rabbits, and this figure by Western blot, we saw that the antibody recognized the SeACov infected viral cells and the purified E. coli expressed purified M protein very well. So we use these antibodies to perform IFA to test the in vitro species tropism of the SeACov. Surprisingly, we found a very diverse species tropism, as you can see here. The Mark 145 from the monkey, the LCPK1, PK15, or ST, and IPAC-J2 cells from pigs, all can be infected by the SeACov. The human cell lines in our lab, HIL7, HeLa, A549, and other human cell lines can also be infected by the SeACov at 24 hours post-infection. Even the hamster cell line, BHK21, which is not infected by most of the coronavirus, can be also infected by the SeACov. However, we have one bad cell line in our lab. This cell line is not susceptible for the SeACov infection. So the results show that the diverse species tropism and efficiency of the SeACov in vitro. So the final update of the epidemiology of the SeACov. So this, the virus-associated diarrhea outbreak has been controlled in these pig farms in Guangdong province by the traditional feedback and the autogenous vaccines treatment. And the most important thing is does the virus spread to the other regions in China? Just 3 weeks ago, the answer is no. However, just 2 weeks ago, a veterinary diagnostic lab in China committed with me. They told me that they can detect the SeACov RNA in a pig farm in Jiangsu province in eastern China. So they sent us the samples and we confirmed their finding. So now the virus does spread. So in summary, a novel swine enteric alphacoronavirus was isolated from the direct pigs in southern China, and this virus is likely antigenetically distinct from PEDV, TGV, and PDCOV. Genomic and phylogenetic analysis show that this virus might have originated from the bad coronavirus HKU2. The extreme entity of the virus as glycoprotein had an extremely high variability compared to that of HKU2, which may be responsible for the cross-species transmission. And the virus is infectious and pathogenic in newborn pigs. So the final conclusion is that this virus represents the fifth porcine coronavirus in addition to the PEDV, TGV, PDCV, and the PHEV. So finally, I would like to thank the founding agency and the 3 graduate students, Yongle Yang, Bin Wang, and Pan Qin, contribute to most of this work. Thank you for your attention.